All right. Welcome, everybody. Hope you guys had a good weekend. Today we're going to be talking a little bit about microbiology, and then I'm going to go over our exams and our assignments that we have going on. Before we get into anything, I just want to pull this sheet up again, show you all this. So I think I brought this up last week to show you guys our assignments and some due dates. I put this up in Canvas under the important links and info tab. And now I've included all the rest of our exams and the dates that those will be on. So if you guys are ever curious about when the next ones are coming up, it's all in here. All 30 points. Remember, your lowest exam grade gets dropped. Important to note, next exam is next week. I know that's really quick, but that's just the way things are working out here. There won't be a ton of material on it, and it shouldn't be too difficult. I'll talk more about that at the end of our lesson today. Cool. Today we're doing microbio. Let's get over here. Sweet. So we're gonna, first I'll talk about exam one a little bit, then we're gonna have our microbio lecture. I'm gonna introduce you to our COVID swab project, talk briefly about our lab manual update, then go over our assignments that are due next week, as well as exam 2, which will be next Monday. First, exam 1. You guys did pretty good from what I saw. Did anybody have any questions, comments, anything they wanted to know or ask about the exam, whether it's material or grading? No? All right. And that's all I have on that. Again, if you didn't do so great, don't worry too much about it. The lowest one will be dropped. Just make sure you do well in the next ones. Cool. Jumping right into molecular biology. This is the central dogma of molecular biology, and what that basically is just an explanation of the flow of genetic information within a biological system. Genes are made up of DNA, and these genes are instructions for making products in your body, and these products are proteins. If you remember, we talked about those a little bit earlier with our biomolecules. If you remember, proteins can be receptors on the surface of your cells. They can also be signalers. Proteins are also enzymes like lactase. Central dogma follows this pattern. So genetic information starts off as DNA. It gets translated into RNA, which is then coding for proteins. In humans, the RNA is an intermediate, and it's messenger RNA. So mRNA is how it's annotated. DNA is found in a membrane-bound nucleus only in eukaryotes. In prokaryotes, DNA is not found in a membrane-bound nucleus. It just kind of floats around. And I'll talk about eukaryotes and prokaryotes a little bit more. Here's just a little diagram of the dogma. So you can see it starts off as DNA with all your typical base pairs. This gets transcripted into mRNA, which then gets translated into proteins. This is a polypeptide, which is just a chain of amino acids that can either be part of or an entire protein. Just another diagram, DNA to RNA to proteins. Oh, too far. We're going to talk a little bit about microorganisms next. These are all going to fall under the category of either prokaryotes or eukaryotes. Prokaryotes are organisms that are made up of cells that do not have a nucleus and do not have organelles with any sort of cell membrane. Eukaryotes are organisms that are made up of cells that do have a nucleus and organelles with cell membranes. So as you can see, you've got nucleus here. All the genetic information is inside of the nucleus. In your prokaryote, you just have your genetic material kind of floating about all willy-nilly without any order. So generally, you're going to see eukaryotes are going to be more complex organisms, prokaryotes less so. First one we're going to talk about are bacteria. These are prokaryotes, so are simpler forms of microorganisms. They tend to be single-celled or unicellular, and they reproduce asexually by binary fission. You guys don't need to know the full process of binary fission. You just need to know that that is how bacteria reproduce. People tend to think of bacteria as disease-causing or pathogenic. They tend to get a bit of a bad rep, but bacteria actually do a lot of good. You have a lot of bacteria in your body. In your gut is probably the most readily available place, and they help you digest all of your food. Cool fact is that in your body, you have 10 times more bacterial cells than you have human cells. So technically, you're only 10% human. The rest of you is bacteria. Our next microorganism is archaea. These are also prokaryotes and all single-celled, unicellular, just like bacteria. They can reproduce asexually as well by binary fission, budding, or fragmentation. Again, you don't need to know how those processes work. You just need to know that that's how they reproduce. They are similar to bacteria, but they don't have a cell wall and they lack peptidoglycans. And those are basically just cell membranes around organelles. So they are incredibly simple organisms. And these are all extremophiles. So an extremophile is any kind of organism that lives in an extreme environment, and this could be extreme temperatures, extreme acidities, extreme pressure. A really common example of an extremophile is a tardigrade. If anybody's ever heard of those, they can live in vacuum, which is pretty wild. But an extremophile, archaea, are methanogens. If you've ever been to a hot spring or any kind of volcanic kind of area, you've probably smelled sulfur, and that is from these methanogens, which are little archaea living in this incredibly extreme environment, which is usually either super hot or really acidic. Our next microorganism is fungi. These are eukaryotes, so these are a little bit more complex organisms. These are made up of cells that have nucleus and organelles. They can be unicellular or multicellular, so you can get bigger fungi than you can archaea or bacteria. And these can reproduce both sexually and asexually. So asexually, they would be reproducing with fragmentation or budding, and then their sexual form of reproduction is via spores, which is how a lot of plants reproduce, a lot of underwater animals and stuff reproduce with spores as well, and that is sexual reproduction. An example of a unicellular fungi is yeast, which we are seeing in our sourdough project. So S. cerevisiae, yep, which is used for making bread and beer. Beer is another really common place you find fungi. Another one is C. albicans, which can cause yeast infections. And this one can switch from unicellular to multicellular. So fungi are not set in either one of those. They can transition. A different multicellular fungi is mold. I know everybody's pretty common or used to mold, and it grows on your bread, but it can also be used to make penicillin. So mold is not all bad. Our next microorganism is parasites. Not all parasites are microorganisms. You can have macroparasites as well. And actually our multicellular down here example is a macroparasite. Another example of a macroparasite would be like a tick. So that's why it's an umbrella term for any organism that lives in or on another organism and benefits by deriving nutrients, usually at the expense of the host. These parasites are almost always eukaryotic. There are a few prokaryotic, but not many. They can be unicellular or multicellular. Obviously, some multicellular ones are animals because it takes an animal. Most reproduce asexually, but they can switch to sexual reproduction. It's kind of cool that they can switch between those two. A unicellular example is malaria, and this is just a small microparasite that infects mosquitoes and humans. I'm sure most of you are familiar with that. And then a multicellular parasite is a tapeworm, and this will infect animals. And again, ticks also a good multicellular parasites. This is just the life cycle of malaria parasite. I'm not going to go through it all right now, but if you want to check it out on the PowerPoints later, go ahead and take a look. It's pretty interesting stuff. Our next microorganism is a virus. Viruses are not actually considered to be living creatures. So the word organism used here is pretty loose. They cannot reproduce. They don't do that at all. They multiply, and they don't do this without using resources from a cell. In humans, viruses will hijack your cell's machinery to make more copies of itself. And they do this using lytic and lysogenic cycles, which I will go over in a second. Some viruses will use only the lytic cycle, but some will use a combination of both, and we'll see how that's possible. And these can be cleared. Some can be cleared from your immune system, but others cannot. So if you've ever heard of HIV, that is a virus that will lay in your system indefinitely. We have no cure for it, so it will always remain there. So these are our lytic and lysogenic cycles. So almost all viruses will undergo this cycle, and others will incorporate both. Either way, it's going to start off like this. You're going to have your cell that has viral DNA inside of it. In this case, we're going to look at it as this little circle. So with the lytic cycle, it transitions from this stage to here, where new viral DNA and proteins are produced and assembled. And these are little virus capsids. Then this transitions up here. The cell lyses, which means it basically just disintegrates, and that releases the viruses out and about. And then they will find another cell and attach to it and inject their DNA. And you start all over. You get your circle DNA, reproduction, development, released, and then starting over. Viruses that undergo the lysogenic cycle do it a little differently. Their DNA will be inserted into the actual cell DNA. Here we're seeing it as a bacterial chromosome. And then as the bacteria divides, that also causes that viral DNA to divide. So then it will get proliferated into the system as that bacteria continues to divide and spread. And then occasionally, the viral DNA will disassociate from the bacterial chromosome, and you'll get back to this stage. And so ones that transition between the lytic cycle and the lysogenic cycle, at least for this graph, kind of run in a figure-8 motion like that. So that's how they get back into the lytic cycle. There are other types of microorganisms beyond what we just discussed. Algae and protists are two big ones. You can also get microplankton. You can also get really big plankton, so that's why we put them in here. But these are important. Algae especially, and I guess algae and phytoplankton collectively are the organisms in the ocean that are generating pretty much all the oxygen in our atmosphere. So super important microorganisms. You won't need to know that for our tests. We're going to talk about managing some of these microorganisms. First, we'll talk about antiseptics versus disinfectants. These are both products used to kill microorganisms, but they do it a little bit differently. Antiseptics will generally destroy or inhibit growth of microorganisms in or on living tissue, while disinfectants destroy or inhibit growth on non-living surfaces. So example of an antiseptic is povidone iodine or betadine. This penetrates into microorganisms and oxidizes the cell's macromolecules, causing them to die. It's pretty brutal, honestly. And then a good example of disinfectant is bleach. Everybody's heard of bleach, and this simply just destroys the cell proteins. Next, we've got our antibiotics and antivirals. Antibiotic, if you just break down that word, it means against life, pretty straightforward. These are used as medication against bacterial infections, and they will typically kill the pathogen itself rather than inhibiting any kind of growth or development. They just straight up kill it. Antivirals are a little bit different. First of all, they're used against viruses, not bacteria. And these inhibit the virus development instead of targeting the virus directly. It's really difficult to actually target and kill a virus. It's hard to do this because the viral DNA is hard to delineate between your own cells. So it's really difficult to target specifically a virus. And this is why we don't have cures for some things like HIV, because it's really hard to do that. This kind of leads into our next parts, which is vaccines. And this is going to be super relevant now, considering we're all taking classes online due to a virus. So you have an innate immune system and an adaptive immune system. Your innate one is the one that you are born with. It works super quickly, but it is non-specific. So an example here is neutrophils, and these are what make up pus. Another innate immune system things like having a fever. It works fast, but it is very non-specific. It can kill a lot of things. An adaptive immunity is the one that you build and develop over time as your immune system learns from previous infection. So this one takes more time to act, but it is much more specific. Example is antibodies. I'm sure everybody here has heard of antibodies. Those will develop as you get diseases. So that's how a vaccine works. Vaccines are a way for your body to learn about pathogens without actually getting sick. So a body is dosed with a very small amount of a disease, and then it will develop antibodies against that disease. So then if you ever catch it again, it knows how to act and how to react to that disease. And we are currently working on a vaccine for COVID right now. Antibiotic resistance. All genetic material is susceptible to changing, and this is commonly referred to as mutating. I'm sure everybody's heard of mutations before. So they also have a bad connotation, but they are not all bad. They allow for us to adapt to changing environments. Mutations are the basis of the theory of evolution. But bacteria and viruses can mutate too, and that causes a lot of issues because they can mutate to adapt to the medications that we use to target them. Bacteria especially can mutate to acquire antibiotic resistance. This is why doctors will always tell you to finish your whole course of antibiotics when you get them, because if you don't, those bacteria will mutate, develop this resistance, and your antibiotics will no longer be effective. You're just going to keep getting sick. So always finish all of your antibiotics and kill those diseases entirely. Cool, that's it for our microbiome stuff. Does anybody have any questions on that material before I go on to our next project? Cool. So our next thing that we're going to be doing is this COVID-19 swab project. And I uploaded the instructions yesterday or 2 days ago onto Canvas. So if you go into Canvas modules under week 6, you will find the COVID swab lab. And I'm going to run through that real quick right now. But you need to pick up a kit. You're going to swab some samples, and then you're going to take the kit back to where it was picked up. That is in San Diego. So if you are not in San Diego, there is an alternate assignment for you. If you guys remember, I asked you all who's in San Diego and who isn't at the beginning of the semester. So I have that list. If your situation has changed and you have left San Diego, please email me and let me know so that we can figure that out. All the details for everything are in the instructions. So we're going to look at those real fast. Cool. Here is our COVID swab. So here's the address. You are going to go to this address in San Diego. If you are in a dorm and you live with a bunch of other people who are in this class and also need to pick up kits, it might be best to choose one person to go and pick up kits for all of you. That way we have less people out and about. You can social distance better. These are available to pick up from 9 a.m. this morning till 6 p.m. in the evening, and they are available till this Friday. So you have to go sometime during this week to pick these up. Please do your best to get that done. Again, if you cannot make that happen or there are some extenuating circumstances preventing you from this, please email me and let me know. If you are quarantined and you've tested positive, you may not participate in this. So again, that would be extenuating circumstances. Please tell me about that. When you go pick these up, wear your masks and social distance. You're going to register these kits online at this address here. So once you've got your kit, you're going to enter the number on there and register yourself. And then what's going to be due next Friday is simply a selfie showing me that you have picked up your kit and your number. I didn't want to make it due Monday because we have a lot of stuff due Monday. There's instructions inside your kits. It's going to be pretty straightforward. You're going to have to take selfies throughout as you're swabbing. I think you have to take 5 swabs, so it's going to be 5 selfies or however amount that you need as you sample. This introduction is going to be all the background on viruses and COVID itself. I would advise you to read over this because I might pull from here as a question on the exam. For your procedure, again, here is the address. Again, available to pick up 9 a.m. today to 6 p.m. all the way till Friday. So you're gonna register your kits. This is gonna be your username and your password. Follow the instructions on the enclosed kits. And for your final write-ups, this won't be due for a while, but you are going to have to create a hypothesis that reflects some thinking about possible viral distribution in your area. And you're going to tie that in with your sampling. So please make sure you read through all of this and complete all of the steps. I will be posting the alternate assignment at the end of this week for those of you who cannot take part in this. If you can, I would highly advise trying to take part in this because the alternate assignment is not like an easy alternative. It's going to be about the same amount of work time-wise, but probably harder brain-wise. It's just a lot of questions. Let's go back to cool, lab manual. So we've got a little bit more info on this. They will not be available in the SDSU bookstore, but I'm going to get a link from the publisher directly, and I will send that to you. You will log on to their site and buy it directly from them. It's gonna be about $20, not including shipping. You'll have to cover that. And it will come with an access code for our upcoming algae project, which I will talk more about next week, but make sure you don't lose that code. That's gonna be important. As soon as I get this link, I will forward it to you and probably post it on Canvas as well so that everybody can get their lab manuals. I know it's a little late in the semester now, but it'll help us with the rest of what we're doing. Next week, you have your 4-week assessment of your first ecosphere due. You have your photo of your second ecosphere setup due, and you have a photo of your second sourdough starter due. Those are all due by midnight next Monday. I know that's a lot in one day, but you didn't have anything due today, so you've had 2 full weeks to work on these. Please get those all in on time. And next Monday, we also have our second exam. And we will have a lecture following it this time. I know we didn't last time, but we will this time. So please show up to class. It's gonna be 35 minutes again. You'll have to log on and have your cameras on at the beginning of class. It'll be roughly 15 questions. Gonna be very similar to our first exam. Remember, it is open note, open book, open internet. Just don't talk to each other. Use any resources you have. Content, we're just gonna go over microbiology and population ecology. Somebody raised their hand. Yeah, do you have a question?
Yeah. You said second sourdough starter?
Yep.
What do you mean by that?
You have to do a second sourdough starter and take a selfie with it.
But when I did mine last week, I submitted 3 of them because I thought we were supposed to do.
No, it was 1 the first time, and then it was very similar to the ecosphere. We're going to do another assessment and hypothesis thing with your second sourdough.
Okay.
All in the instructions, yeah. And I talked about that last week as well. Back here, so it's going to be, our lecture's going to be, or exam's going to be on microbiome and population ecology, so just 2 lectures. And potentially there will be a question from those COVID instructions, so make sure you read through those. That's it. Cool. Does anybody have any questions on material, assignments, or exams? No? Yeah, Sebastian?
So I looked up the address for the kit, and it shows a house. Is that correct, or?
Yep, it is somebody's house who's helping us with this project. Oh, important note on that. Actually, I just got a question from my last class. It's going to come in a cooler. Make sure you refrigerate that contents when you get back. Some of the stuff in there needs to stay cool. Any other questions on anything we're doing? All right, then. That's it for today. I'll get the PowerPoint recording up on Blackboard by the end of the day. So you guys are free to go.